Hi, this is Brianna Hunter, a postdoctoral fellow at the UC Davis Center for Mind and Brain. I'm excited to share our recent work to be published in Developmental Science entitled Relating Infant Fixations to Adult Cortical Activation Patterns Using the Natural Scenes Dataset. Visual attention is one of the earliest tools infants use to explore the world. The ability to control eye gaze is therefore a foundational skill that sets the stage for much of what infants learn. Traditionally, research has shown that young infants' gaze is captured by simple visual features like brightness and contrast. But across the first postnatal year, their attention becomes more adult-like and guided by increasingly meaningful features of the environment, such as faces or objects. This progression from simple to abstract mirrors the architecture of the visual system in the brain, in which light enters through the retina and is processed first based on basic visual features in early visual cortical regions, but becomes increasingly abstract as this information moves through later cortical regions. What we've been missing is a direct way to link infants' gaze patterns to the kinds of visual representations coded in the different regions in the brain. This gap has limited our understanding of how specific cortical structures support infants' visual exploration. Our team tested 93 infants split into younger and older groups, plus a comparison sample of adults. We showed them a series of up to 50 photographs of common places such as living rooms, restaurants, and parks. We created fixation density maps, like this one pictured on the right, that reflected where each subject looked while viewing each scene. To relate these gaze patterns to the brain, we leveraged a large open fMRI dataset known as the NSD. In this dataset, 8 adults spent nearly 40 hours in a high-resolution MRI scanner viewing thousands of naturalistic scenes like those shown to our subjects, yielding high-quality data demonstrating precisely how these scenes activate distinct cortical regions. We then used a technique called representational similarity analysis to relate our fixation data to the fMRI data in the NSD. First, we correlated infant fixation density maps for each of the scenes viewed by each subject. This allowed us to generate representational similarity matrices, or RSMs, that show how similar gaze was for each scene viewed by each subject. We did the same for activation across voxels in the fMRI data from the NSD, separately for 8 different regions of interest, or ROIs, that were already delineated within the NSD. Representational similarity was defined as the rank correlation between the fixation RSMs and the fMRI RSMs, where representational similarity significantly greater than 0 indicates a significant link between the two data sources. Although we explored 8 different ROIs in our paper, in this video I'll present the results from 3 of the ROIs that we are primarily interested in, which span from low levels of the ventral visual stream up through higher level areas. Using this method, we found that younger infants' fixations were only significantly related to activity in low-level visual areas, regions that process basic features like edges and orientation. However, older infants' fixations were significantly related both to low-level areas and to mid-level visual regions, which process more complex features. This pattern was similar to the results from our adult subjects, suggesting that older infants' gaze was more adult-like. In summary, across the first year, infants' attention shifted from being guided mainly by simple visual features to being increasingly shaped by more abstract representations closer to those used by adults. Our findings suggest that higher order visual areas may play an increasing role in guiding visual attention across infancy, reflecting a developmental shift towards a more abstract representation of the environment. These findings help us gain insight into how infants learn to represent the world, with important implications for documenting both typical and atypical trajectories. To learn more, please check out our paper in Developmental Science. I'd love to hear your thoughts or questions, so feel free to reach out to me at bkhunter@ucdavis.edu. Thanks for watching.